join us in Zoom, we kindly ask you to write your questions down in the chat so that the speaker can answer them in the corresponding section of the talk. We kindly ask you to put your mobile devices on silence for the duration of the conference. Thank you very much and welcome. Today, we will be receiving Research Professor of Simvestat Irapuato Unit, Mexico, postdoctoral researcher, University of California, Berkeley, USA, our postdoctoral researcher and European Molecular Biology Laboratory in Germany, PhD in Natural Science, Albert Karl University Heidelberg and European Molecular Biology Laboratory in Germany, and the Bachelor's in Degree in Biology, National Autonomous University in Mexico, Dr. Kasia Oktaba, coming from Irapuato. Please take a big round of applause to receive her.
Thank you very much. It's a great pleasure to be here live and alive because I really apologize because of the delay that is starting. But yes, the highway, if you have ever come from Irapuato to Querétaro, can be a pain, and unfortunately today it was fixed. But without further ado, today I'm going to present you what we do in my lab in Simvestat Irapuato, and the title of the talk is Genome Topology in Drosophyla, and I'm going to explain you what's this genome topology. So thank you very much, Diego, for inviting me and for the nice introduction. So just to start with, wasn't it this one? Sorry, I misunderstood something.
No, it's just you have to click on it to click somewhere. Right.
Okay. So just to start with, since the last years we have been starting to sequence each time more and more genomes. We have now quite a lot of them, and here I'm showing you like some of the most important model organisms that we work with in general in biology, and maybe you can appreciate that the sizes of these genomes that are shown here are very diverse. So we have, let's say, smaller genomes and larger genomes as our 3.5 bases. However, the number of genes contained in these genomes is relatively in the same range, so plus minus 20,000 genes. So what we are thinking of this is that since there are so many different phenotypes in this diverse world, it's not really the genes that are coded in these genomes, but the sequences that are between the genes, which are making these huge differences in the different genomes, that are important for the regulation of these genes that we share among the organisms, and this different regulation between these organisms gives us these very different phenotypes that we can see in nature. So differences in how genes are regulated are those that are giving us these diverse genomes. So on the other hand, if we think of the cells, each one of the cells that build each one of these organisms, so each cell in our body has exactly the same genome, right? The same, in principle, the same DNA sequence. And nevertheless, they are very different, the cells that constitute our body. So you can have cells like this one, which are astrocytes from the nervous system, or cells like this one, which are skin cells. And as you can see, they are very, very different. Yet the genome that these nuclei have and the genome that these nuclei have is in principle the same. So there should be also differences in how these genes are regulated. Some are silent, some are expressed, so that we have these different phenotypes in our tissues. So in our lab, these are the questions, and I will come back to this in our talk. These are the questions that drive our research. how these different cells that form different tissues on animal bodies get there. So how differential gene expression is happening so that we have these different cell types and different phenotypes in nature. So we do not study human cells. We do our experiments in Drosophyla, the fruit fly that you can find sometimes in the kitchen with red eyes. And here this is a Drosophyla embryo, a lateral view, and this is a ventral view. But it doesn't matter too much. What I wanted to show you here is that if you can see, each one of these dots is a cell. And at this time point in development, this is an early embryo, this is a later embryo. Drosophyla development takes about 1 day to happen from embryo to larva, which is the next developmental stage. And each one of these cells is expressing different genes. So there are some cells that express the red gene, there are some cells that express the pink one or the yellow one, but it's not that all cells express all the genes at the same time. And this differential pattern of expression is very important because, for example, these yellow cells are part of the nervous system of the Drosophyla, and only those need to express those genes and not the dermis or. So in our model of study, we exactly look at these expression patterns, and we want to know how these cells do not express the green gene, and these cells do express. And why Drosophyla? Well, when I was doing my PhD, I got into contact with Drosophyla, and until today, I'm using this model organism. I think it's a very nice organism to work with in a lab. It doesn't take too much space, and actually, it looks very different from a human, and we diverged 600 million years ago. However, 60% of fly genes have human homologs, so actually we are not so different. And 75% of genes responsible for human diseases have an homolog in flies. So if we understand the mechanisms of gene regulation in the fly, we can also get to know more on how this gene regulation happens in humans. So there are like two aspects in my lab that we study. On one hand, we study if you see this is like a fragment of the genome, so it's a DNA sequence, and you have a gene coded there. As I told you, there are some regions that are regulating this gene expression. So we're interested in knowing which are these regulatory regions, because they are not so well annotated as genes, and where are they in the genome, in these huge, sometimes genomes that animals or organisms have. On the other hand, actually the DNA, it's not like this in the nucleus. It's not a linear DNA sequence. This DNA is arranged in the nuclei in a specific way, and this also has an impact in how genes are expressed and where. So these are like the two main aspects in my lab, and genome topology is exactly this, how DNA is arranged in the space, the 3D space of the nuclei. So to start with the identification of regulatory regions, what we do is we do experiments like, for example, ChIP-seq. I don't know if you have heard about ChIP-seq, but what it's all about is to have antibodies that are specific for a transcription factor or an epigenetic mark or something that is of interest. And in our case, it's an epigenetic mark, which is the acetylation of lysine 27 of histone 3 that is in the nucleosomes that wrap DNA around. So we use antibodies against this mark because we know that this mark is present at regulatory regions. Not at genes, not in other places, but at regulatory regions. And this helps us to identify in a whole vast genome where these regions are, like knowing where this epigenetic mark is located. So these are results from our lab, and each one of these is a track, and this is from different tissues that I'm labeling here, A, B, C, color-coded, and maybe the details doesn't matter, but you can maybe recognize that there's like more signal or more color in this region. And that is because this region is packed with regulatory regions. And this is how we identify them, and we then map them to the regions in the genome, and we say, okay, this is interesting. Here, there's something happening, and this might be regulatory of the genes that are nearby. And we also do experiments like RNA-seq, where we know the readout of these genes, right? So how genes are expressed. So in the different tissues, the same ABC, we know that in tissue C, this particular gene is expressed. So here you have zinc, and in the other tissues, B is not expressed, in C, it's not expressed. So this regulation of this pink region, looking at this gene, is what drives the expression of the genes at that region. And it's only happening in this tissue and not the other two. So in that way, we identify these regulatory regions or enhancers, and then we sometimes clone them with reporters so we can see the expression that these enhancers drive in the embryo. Because we cannot see all the genes by eye, we use reporters. And for example, we have identified that many of the developmental genes, so those genes that are involved in the development of Drosophyla, do have more than one regulatory region. And so many enhancers, call them enhancers, and some of these enhancers work redundantly. So, for example, the light green enhancer and the darker green enhancer, they both can drive expression in the same regions. But there are two, and this makes the system very robust, because if evolution wants to explore and get new expression patterns in the embryo so that it can evolve differently, then if this is redundant and both enhancers give the same expression pattern, then evolution can start mutating one of these enhancers, and then maybe we can get new regulatory regions, new expression patterns, and new phenotypes. So that was how we identify and then we verify that regulatory regions have a function and regulate the expression of genes. But then the other big part is this genome topology, which is like leaving behind that this genome is like a string of DNA, but looking at how this genome organizes in the nuclei of the cell and how this impacts this gene regulation. So for instance, our genome, like this 3.5 gigabases, is like, if we put it like this, completely unfolded, it will be 2 meters of DNA, a bit more than that. And then each of these 2 meters needs to get into the nuclei of your cells and get arranged there and compact, because our cells are 5 or so meters. And I think here I have, this is guinea pig, but anyway, it's DNA that needs to be arranged in the nuclei of our cells, and 2 meters of sequence has to go into a 5 to 10 micrometers sphere that is the nuclei. So you can imagine it's a lot of work to compact that DNA, and still this DNA needs to be functional, right? Still, if you need to express one gene at what point in one tissue, then this gene needs to be available in all this mess of DNA in that nuclei to be expressed and to be regulated correctly so that this organism develops well. So we have known that the nuclei here is a big guinea pig bone marrow cell, nuclei from 66, so quite already some time ago. And this picture already shows that in the nuclei, the DNA is not arranged like very homogeneously, but there are regions where chromatin is more compact and some other regions where chromatin is more lax. So this has been known since long time. And we have known that if you look more closely to DNA, it arranges around these nucleosomes that I already mentioned and forms these bits of a string, and then it's more compacted and forms these fibers of 13 nanometers. This is what textbooks always tell us. And the thing is that this is ex vivo, so we can see it, and it happens, but this is not in live cells. If we really look at cells in vivo, then we can see that it's rather something like this. So it's but it's never such maybe a nice ordered chromatin, but it's something like this. So it's a disordered 524 nanometer chain that sits in those nuclei. So this is the basis, like the very basic arrangement of DNA in nucleosomes, and you have heard about it. But now, with all the development of different techniques, you can study how this genome arranges in the nuclei at different scales. So what I was showing you, it's more or less this scale of nucleosome, and this is DNA. But now you can look at other levels of organization, like loops and tags, something that I will come back to, and then even look at organelles and how this DNA rearranges the cells. So our lab is focused at this level, doing some experiments that are called Hi-C. So Hi-C, I don't know if you have heard about this technique, but Hi-C is a molecular biology technique, and we do it all the time in the lab. And it's all about basically cutting and pasting the genome that some cells of interest in Drosophyla have for us. And then we sequence this massively. Many of the fragments that had an arrangement in the 3D space of the nucleus, we process data bioinformatically, and we get into these matrices that we start interpreting as, so like these triangles, those are regions that are full of contacts, which means that DNA there contacts itself a lot, or something like this. And then there is a region where you don't have so many contacts, so red is a lot of contact interaction, and white is no contact. So we have these kind of triangles, and there are a lot of contacts here, but then there are no contacts, and again another triangle that is smaller, and so on. And then we start taking all this information and building it into how this represents what is really in the nucleus. So this is an overview of this genome topology because it has different scales. So at the highest level, let's see, or levels, are the chromosome territories. So everybody has heard about chromosomes, right? Those are also packed in the nucleus. So each of the chromosomes has its own space. So it's not that the chromosomes mingle around in this nucleus. And we see that in our matrices, like these triangles of high interaction. So this is all one chromosome that is packed, for example, this, let's say, here. Then in the next level, if we look at one of these chromosomes, we start doing a zoom, we can start observing different patterns. And these patterns just correspond, as we saw in the nucleus of the guinea pig, like regions of high compacted chromatin and others like the yellow that are less compacted. Then, if we look even closer to this chromosome, we can see these triangles that are the different topologically associating domains. So again, regions of a lot of contacts, like these, that are separated by lower amount of contacts. And then there's another tag, so another topologically associating domain here that is separated by a region of no contacts, and so on and so forth. So these topologically associating domains are of great interest for us because one of these domains encompasses like the regulation of different genes that are located there. And it makes that the regulatory regions, these enhancers, for example, are located in here and interacting with the genes that are here, and not contacting and regulating genes that are nearby in the next step. So all the regulation seems to be, or most of it, happening in one of these tags, and the genes that are contained there are well regulated thanks to that structure that keeps that environment tight for regulation. And they do not mix with the next step that is just besides. This one has their own genes there inside and regulating those, and there's no cross-regulation or not much between these bodies or tabs. And then if we get into one of these tabs and we look even closer, then we have these kind of loops. So these are really regulatory regions that contact the promoters of genes, as we saw in the slide, so that are regulating, regulatory regions regulating target genes. So this is genome topology, and when we do our high-C experiments, we ask what's the topology in different tissues, and we start analyzing each of these levels to try to understand at each of these levels what's happening in our tissue of interest. And this is kind of what is shown here. So these are also results from our lab, where you can see this is a zoom to different regions of this genome in, again, tissue A, the green tissue, and tissue C. So as you can see, this is the same region, and this is the same region, and this is the same region. And as you can see, this topology varies if we focus on this case. So in one tissue, maybe it's not so evident, but here there is some lack of contact that separates these triangles. However, in tissue C, it's one big triangle. So we are interested in spotting these differences that reflect the topology that is tissue specific. And this correlates well with the expression of genes that are located in this region. So for example, tissue A expresses a gene which tissue C doesn't express. So this is gene active, and this is the topology when the gene is silent. So we look at differences in the topology and how this DNA is arranged in these nuclei, and then look at how this correlates with how genes in that region are expressed. And we do this tissue specifically. And then we are also interested in ones that we know how does the topology look like in the wild-type situation in that tissue C or A or B. We start to play with that topology. So we start deleting regions that we think, for example, the triangle seems to finish here. So we delete this region with CRISPR-Cas, for example, editing systems. I don't know if you have heard about it, but it's a very nice system to delete regions very precisely of the genome. So we delete regions that we think that are important for the organization of this topology, and we look at what's the result on the expression of the genes that are around or the phenotype. You remember at the beginning I told you this regulation of genes is all what gives us the different phenotypes in nature. So we
Professor?
So we start modifying and editing these elements that we consider that are important for establishing this topology, and we look for phenotypes. So this is the wing of the fly. So if the development of that embryo that I showed you goes well, then you have a larvae, and then you have an adult, and this adult fly has a head, and antenna, and legs, and wings, and all is fine. But if at some point of development, the regulation of the genes is altered, or the topology is changed, and that has an impact in the expression of that gene, then the phenotypes can change. And this is what you see here. So this is a wild-type wing of our Drosophyla. And when you start modifying this topology, then you have phenotypes where you can recognize that the genes were not expressed correctly in this region, that will change, and something is going wrong in development. And these poor flies have these wings that maybe are not as functional or whatever than the wild type condition. Okay, so what we know so far, and in the next couple of minutes, what I want to emphasize. So the other ones are the kind of experiments that we do in our lab. So identifying regulatory regions in this one genome, or trying to identify the topology, how does the genome arranges in space, and how does this impact the regulation of the genes. So, and once we have this information, we can start to think about the mechanisms of how this topology works, or how well the mechanisms are conserved among different organisms, or at all if there is topology in every living organism, or it is something specific. So, of course, we know that no, that's not the case. So, and we start thinking about mechanisms. So we know, I will tell you like very general things that we know so far about genome topology and how it works. So in order to have this, what I think, but maybe I'm biased, very nice expression patterns in Drosophyla. You see the cells and the colors, and they are different. So in order to get these different colors in this embryo and in other organisms, you need a very precise regulation in time and space. So what I mean is in space, well, you need to express the red gene in this stripe, and this is important because this will allow the fly to have different segments. And then from some segments, you will have flat wings, and from other segments, you will have the head. And so if you take some of these cells and you look at the expression of the red gene, you will find something like this. So the topology, this is a silent case where the genes are kind of close in space in the nuclei, but they are not being expressed. However, if you look at the stripe, at cells in this stripe, you will look at the expression of the red gene, let's say it's gene 2. And you will see that the topology changed a bit in this loop of regulation. And now you have what we now call transcription hubs, where you have like an environment that is very nice for genes to be active. meaning that all the transcription factors that regulate this expression are around the blue bubbles. And you have the polymerase that needs to transcribe, and you have some other general transcription factors, and all is there in this transcription hub, making it possible that the genes are active. And when the gene is not expressed, then all this hub is not happening. So the other thing that is still, I think, a bit missing in genome topology is that I show you these 2D red and white matrices. And I told you that we are interpreting this topology from those matrices. And well, the genome is arranged in a 3D space, and these matrices are just 2D. So we need still to get a step further and analyze the topology. These are nice experiments, but we're not doing this in the lab, but looking at this, doing these high-C experiments in different nuclei, in different cells during time, so that we can construct from this really. And from our experiments, we could also do it. But I think this is the lacking or not so developed still part of genome topology, to try to get to these models that really show the genome in the 3D space of the cells. So what we really know is that basically any organism that you look into has some kind of topology. So this is a representation from a very nice paper from 2 years ago, and there are, as you can see, different organisms, plants, and you have fungi, and you have flies, and fish, and chicken, and all of them have topology. So, of course, the genome in each of the cells organized somehow. And then if you have all this info, or how this happens in the different organisms, you want to start looking at what are the similarities and what are the differences. And we have started to understand if the way this genome is organized in each one of these organisms is the same, are the mechanisms the same, or it is very different, or if some use some kind of proteins and others use other kind of proteins to organize this genome. What is conserved, what is not conserved, what are novelties in evolution. So these are also like very much what is happening now, that we are comparing how different organisms organize their genome in the nuclei. And then, for example, if we look at Drosophyla and vertebrates, in vertebrates, one of the proteins that we know it's like the most important, or the protein that organizes the genome into these triangles that I have shown you, it's called CTCA. It's like an architectural protein that works for that. And we know that in Drosophyla, well, if you can, do you see any difference between this and this? Yeah, probably no. Of course, it's difficult to just get about somewhere. But the answer is no, there's not much differences when in Drosophyla you take out this protein. So clearly, this mechanism of organization of the topology in humans is a bit different from Drosophyla. And this is because Drosophyla has a lot of other architectural proteins that do the job. So whenever we don't have CTCA, there's another protein that enters there and arranges this topology. And in humans, it's just this CTCA. We know so far that it's doing all this. So there are differences in the proteins that arrange the topology when you look at different organisms. And then, well, is it relevant? Is this topology really important? Well, of course, I would say yes, but maybe this convinces you. So there are many syndromes in humans. as you know, or diseases. And some of those have been associated to aberrant topology, genome topology. So these are nice experiments where you alter this topology, as we do in our experiments in the lab in Drosophyla. We start deleting regions that we think are important for regulation, and then so changing the topology of a specific locus can give this aberrant phenotype. So it seems that it is important to keep this gene regulation very tight and very nice in space and time in the different tissues, and to have this topology as a frame, like a scaffold, so that this gene regulation happens really properly, so that we do not have these aberrant phenotypes that can cause disease. Okay. So with this, so that we can have some questions, I want to finish and show you my lab. This is actually on Monday when Liz graduated. She did her master's at my lab. And this is Alejandro. Eduardo, I showed you some data from him, and Diego was working with him for some time. There's Diana, Liz, Joanna, who is my right hand, auxiliar, and Fernando, a postdoc, and Gabriel, also a master's student. So in Irapuato, we have, this is my email. If you have any further questions, please do not hesitate to contact me. But in Irapuato, we have 2 graduate programs. So for master students and PhD, Biotecnología de Plantas y Biología Integrativa. So if you are interested in this or other biological questions or evolutionary questions, well, just check what we are doing, and you're very welcome to do your postgraduate student studies with us. And of course, we are funded by the Institute, partially, but also by Kasia, who funds our research. So with that, I thank you very much.
Thanks a lot. It was a very interesting topic. I really personally enjoyed it.
Maybe I speed it up because I thought you were late, and then now, if you have any questions, please ask. But if you don't, I hope you enjoy it.
Before my question, I want to be this part of the university, and thanks for coming.
All right, thank you very much. That's very nice for me.
If any have any questions, we may address. Maybe
anything. It can be science in general, women in science, whatever you want. It doesn't have to be about topology. Go ahead, yes.
So you mentioned some experiments using different tissue types. Were those exclusively embryogenesis tissues, or have you looked at that in adult flies that might, be regulating gene expression differently, particularly having to do with aging, for example?
Yeah, that's a very nice question. So we do. I'm not very fond of development as a process. I do not consider myself as a developmental biologist. I do like to use development as a readout of gene expression. So what I like is regulation of gene expression. For example, it can be a fly. I have projects in plants. I have projects in lizards. So different, but yeah, my lab focuses on plants. But so gene regulation is happening everywhere, and we use the development of Drosophyla as a model. So we have focused in embryogenesis and embryos because it's easy to have lots of materials, lots of biological material from the embryos, because one female fly can lay up to 100 or more eggs a day. So we can have a lot of embryos to study and to do our questions. Then sometimes we look at adults, like the flies, like the wings I showed you. So we sometimes follow development of the larval stages or even adult tissues as the wings, because it's easy also to look at wings and spot phenotypes. We had a project about aging with a very nice colleague, Alexander de Luna, in the staff. And he studies yeast and aging in yeast. And we were looking at some genes that they discovered that has to do with life being longer, lifespan, longer lifespans. And we did some things in Drosophyla to see if the mechanism again is the same and so on. But it was a collaboration, and I really do not focus on aging, but I use development as a bit out. It's interesting, but I like embryos.
You mentioned that there are other architectural proteins.
Can you speak a bit louder?
Yes. You mentioned that there are other architectural proteins in Drosophyla. Are there any studies currently being made on those other architectural proteins?
In human
or in Drosophyla?
So yeah, we have those are proteins that have been known actually for quite a long time. And they have been categorized usually as insulators. And we have suppressor of heroin, we have CP190, we have P32, we have CTCF, of course. So there are many, there are 6 that are the most common ones. And yeah, we know that sometimes they work redundantly. So when CTCF is not there, and in Drosophyla, CTCF has been related more with the nervous system. Somehow CTCF likes to organize the nervous system. But other than that, there are the other proteins that sometimes we have 2 or 3 at the same place, or 6 at the same place, organizing this topology. So it's a much more, there are more proteins doing this job than in humans.
Questions? Well, thanks a lot for coming to you all. And let's give another big applause to Dr. Kasia Oktaba. Our next talk is at 1 p.m. in Insights in the Pharmaceutical Industry. Who wants to stay? And if not, Dr. Kasia will be here in campus a little bit more time if anyone have any questions, even about internships or social service.
Sure. Thank you very much. Nice campus, by the way. I hope I can see more.